Hello everyone, today I'm going to tell you how the glycolysis, Krebs cycles, electron transport chain perform in our cells. So we can see from the diagram here, glycolysis occurs in the cytosol. The pyruvate, which is the end product of the glycolysis, will combine with the coenzyme A to form the acetyl-CoA and then enters the Krebs cycle. The Krebs cycle occurs inside the mitochondrion. After the Krebs cycles, the electron transport chain will occur in the intermembrane space of mitochondrion. First, what is glycolysis? Glycolysis is a series of reactions that extract energy from glucose by splitting it into two 3-carbon molecules called pyruvate. It transfers energy in the sugar molecule to ATP. In organisms that perform cellular respiration, glycolysis is the first stage of this process. A phosphate group is transferred from ATP to glucose, making glucose 6-phosphate with the help of hexokinase. And glucose 6-phosphate is converted into its isomer, fructose 6-phosphate, with the help of phosphoglucoisomerase. A phosphate group is transferred from ATP to fructose 6-phosphate, producing fructose 1,6-biphosphate. This step is catalyzed by the enzyme phosphofructokinase. Fructose 1,6-biphosphate splits to form two 3-carbon sugars, which are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. They are isomers of each other, but only one. Glyceraldehyde 3-phosphate can directly continue through the next steps of glycolysis. Dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. Two half reactions occur simultaneously. The first one is glyceraldehyde 3-phosphate is oxidized, and the second one is NAD+ is reduced to the NADH and H+. The overall reaction is exergonic to phosphorylate the molecule, forming 1,3-biphosphate glyceride. 1,3-biphosphate glyceride donates one of its phosphate group to ATP, making a molecule of ATP, and turning into 3-phosphoglycerate in the process. 3-phosphoglycerate is converted into its isomer, 2-phosphoglycerate, with the help of phosphoglyceromutase. 2-phosphoglycerate loses a molecule of water, becoming phosphoenolpyruvate (PEP). This PEP is an unstable molecule, which tends to lose its phosphate group to ADP, making a second molecule of ATP. As it loses its phosphate, PEP is converted to pyruvate, the end product of glycolysis. So now let us proceed to the Krebs cycle. Krebs cycles take place in the matrix of the mitochondrion. Unlike glycolysis, the Krebs cycle is a closed loop. The last part of the pathway regenerates the compound used in the first stage. So now we come to the first step of the Krebs cycle. The first step is a condensation step, combining the 2-carbon acetyl group from acetyl-CoA with a 4-carbon oxalacetate molecule to form a 6-carbon molecule of citrate. Citrate loses one water molecule and gains another as citrate is converted into its isomer, isocitrate. Isocitrate is oxidized, producing a 5-carbon molecule alpha-ketoglutarate, together with a molecule of carbon dioxide and 2 electrons, which reduce the NAD+ to NADH. Coenzyme A combines with the succinyl group to form succinyl-CoA. Steps 3 and 4 are both oxidation and decarboxylation steps, which release electrons that reduce NAD+ to NADH. and release carboxyl group that forms carbon dioxide molecule. A phosphate group is substituted for coenzyme A and a high-energy bond is formed. This energy is used in substrate-level phosphorylation to form either guanine triphosphate or ATP. The product in this stage is succinate. Step 6 is a dehydration process that converts succinate into fumarate. Two hydrogen atoms are transferred to FAD, producing FADH2. The energy contained in the electron of these atoms is insufficient to reduce NAD+, but adequate to reduce FAD. Water is added to the fumarate during step 7 and malate is produced. The last step in the citric acid cycle regenerates oxalic acetate by oxidized malate. Another molecule of NADH is produced. The cycle starts again. The products of Krebs cycles. Each turn of the cycle forms 3 NADH molecules and 1 FADH2. These carriers will connect with the last portion of aerobic respiration to produce ATP molecule, and 1 GTP or ATP is also made in each cycle. Lastly, we come to the electron transport chain. Electron transport chain has 3 steps. The first step is generating a proton motive force. The hydrogen carriers NADH and FADH2 are oxidized and release high-energy electrons and protons. The electrons are transferred to the electron transport chain, which consists of several transmembrane carrier proteins. As the electron passes through the chain, they lose energy, which is used by the chain to pump protons from the matrix. The accumulation of the proton within the intermembrane space creates an electrochemical gradient. Then we come to the step 2, ATP synthesis via chemiosmosis. The proton motive force will cause the hydrogen ion to move down their electrochemical gradient and diffuse back into matrix. This diffusion of protons is called chemiosmosis and is facilitated by the transmembrane enzyme ATP synthase. As the hydrogen ion moves through the ATP synthase, they trigger the molecular rotation of the enzyme synthesis ATP. The step 3 is reduction of oxygen. In order for electron transport chain to continue functioning, the de-energized electron must be removed. Oxygen acts as the final electron acceptor, removing the de-energized electron to prevent the chain from becoming blocked. Oxygen also binds with the free protons in the matrix to form water. To remove the matrix protons, maintaining the hydrogen gradient. In the absence of oxygen, hydrogen carrier cannot transfer energized electron to the chain and ATP production is halted.